treadmilling. Most of you must have used a treadmill or at least seen one. When you're using a treadmill, you're walking or you're running, but you're going nowhere. It's sort of the same principle. Let me explain this with the help of this pen. If this pen were a dynamic structure, meaning it could change, add or become shorter or longer, imagine a scenario where this pen was getting shorter at this end and longer at this end. Okay, so you're losing subunits from this side, but you're adding subunits at this side. If both the rate of addition of subunits and rate of loss of subunits was equal and the same, you're losing same amount of monomer subunits as you're adding, then ultimately what will happen, if it gets shorter here and if this pen gets longer here, it will appear that this pen is actually moving. In actual fact, this pen is not moving. It is just getting shorter at this end and getting longer at this end. Let me explain it to you with the help of certain diagrams. And also, I'll show you an animation in the next module. But for this module, let's talk about filament treadmilling first. Actin and tubulin subunits are also enzymes that can catalyze hydrolysis of nucleoside triphosphate, ATP or GTP. We know that ATP is attached to actin monomers and GTP is attached to tubulin monomers. So hydrolysis proceeds very slowly in free subunits, but the rate of hydrolysis increases significantly after the subunit has been incorporated into the polymer or the filaments. Free phosphate group is released from each subunit, but the nucleotide diphosphate remains trapped in the filament. So if we are talking about actin, ATP will be hydrolyzed and a phosphate group will be released, but the ADP will still be part of the actin filament. When the nucleotide is hydrolyzed, where does the free energy go? We have said that it is a high energy bond. The energy which is released from the hydrolysis is basically stored in the lattice structure of the filament, be it microtubule or actin. The equilibrium constant for dissociation, Kd, we are symbolizing that with Kd equals K off divided by K on. For the D form of the polymer, which is equal to the numerical value of critical concentration, which is this concentration is greater than the equilibrium constant for the T form. Let me actually take you to a different slide where I can explain this to you graphically. I think it will be easier. Here, for example, we are looking at a slide in which monomers are coming together and forming a polymer. So the soluble subunits in T form are this square. The polymers, when the monomers come together and form the polymers, they can hydrolyze their GTP or ATP. So the T form of the monomer is this box, and the D form of the monomer is this circle. So polymerization occurs. You form the filament. As we have said, the rate of the hydrolysis is slow. So slowly the individual monomer units will start hydrolyzing their ATP molecule. For the sake of simplicity, let's just talk about actin, so there's no confusion about ATP or GTP. So we are looking at actin filament. So this monomer has in the actin filament has hydrolyzed ATP into ADP. And these monomers which are added at a later point they still have their ATP. The subunits keep on attaching, adding on the plus end, and at the very tip you have monomers which are in T form of the polymer. At the minus end you have the monomers which are in the D form of the polymer. So keep this in mind when we are talking about the other information on the other slides. I'm going to go back and we are going to talk about some of the information I skipped there. So let's talk about this now. So addition of critical concentration, the amount of monomer required to add on to the D part of the monomer, remember the circles, is greater than the critical concentration at the plus end where the T form of the monomer is present. T form of the monomer is present in the polymer. Okay, so CCD, critical concentration at the D part of the polymer, and critical concentration at T form. The D form is greater than the critical concentration for D form is greater than the T form, meaning that basically you need higher concentration of monomer to add on the D form or the minus part of the polymer. Let me go back to that slide again, if you allow me here. So you need higher concentration of monomer if you wanted to add subunits here. The addition of subunits can occur at a lower concentration at the plus end compared to the minus end. That's basically what that statement says. I hope it is clear to you. Now let's go back to our original slide. At a certain concentration of free subunits, D form of polymers will shrink while T form of the polymers will grow. Remember, there is a difference of critical concentration between the two ends of our polymer. So in living cells, most of the free subunits are in T form. Concentration of ADP and GTP is higher in the cell. The longer subunits are in a polymer lattice, the more likely they have to have hydrolyzed their nucleotide, which is bound to the monomers. Relative rate of hydrolysis and subunit dictate whether the filament tip has subunits in T form or D form. So if the rate of addition of monomer is greater than the rate of hydrolysis of the ATP, for example, the plus end tip of your filament will be in T form. If the rate of hydrolysis is slower than the addition of the rate of addition of monomers, then your tip at the plus end will be in T form. So we have talked about this already. So let me show you this graph at a certain critical concentration. This is basically elongation rate, and this is on the x-axis you have subunit concentration. So as you increase the subunit concentration at the plus end critical concentration, now the concentration of subunit increases, and at the plus end you start to elongate. So you are elongating. Whereas at the minus end you need a higher concentration. This is the concentration of subunits you need to start polymerizing at the minus end of the filament. Okay, so between these two subunits you have in this range. If your concentration of monomer lies in between these 2 points, you will basically be adding subunits at the plus end of the filament, but you will be losing monomer from the minus end of the filament. See, before the elongation rate, below 0 means it is getting shorter. So if we have our concentration of our monomers is somewhere here, you will be basically adding subunits at the plus end and losing at the minus end. Graphically, if you look at this in a form of in pictures, let me show you pictures of this. So here you have a filament. This is basically tubulin, which has been labeled with a fluorochrome. You make a mark on this microtubule. This green structure is a microtubule. You make a mark on it and you follow this. So the mark stays at the same spot, meaning if the mark hasn't moved, this microtubule has not moved. If the mark had moved, we would have said the microtubule has moved. So mark stays put, but see, this filament seems to be going in this direction, and because it is getting shorter in this part and it is getting longer in this part. Again, if you follow the sequence of photographs, you can see the treadmilling very nicely, that you will see that filament, although it is stationary, the microtubule itself is not moving. However, it is getting shorter at one end and longer at another end, giving an illusion that microtubule is moving. Microtubules, by the way, do move, but the photograph I showed you was that of treadmilling. Treadmilling is losing monomers at one end, addition of monomers at the polymer. At the other end, both the rates are about the same. It seems that your polymer is moving. In fact, it is not.